All right, hi there. So, up to this point, we've been talking about Darwin and his theory of natural selection as a means for evolution. Now, we're going to be talking about evolution itself, and we're going to be talking about the evidence that supports the concept of evolution. And there are several different ways that we are going to be able to, several different pieces of evidence that we're going to be able to look at. So, let me share my screen with you. So, evolution. What is there that supports this idea that evolution has occurred? What evidence is there? So, evidence number one, the first piece of evidence that we can look at is the fossil record. The fossil record is not complete. There are lots of missing pieces, but we are able to look at the remains of species and see how those things have changed over time. So, here we can see the changes that have occurred in the skulls of the Homo sapien over time, and that allows us to understand the evolution of our own species. However, we can't really do this if we don't know which one of these pieces is older or younger than the other. So we need to first be able to put them into a sequential order in order to see how they've changed over time. So we can do this in one of two ways. We can use a process called relative age dating, where we compare the relative ages of rocks in a rock layer and the fossils within them. Or we could do something called absolute age dating, which actually gives us a number in years. So let's take a look at these two methods. So first off is relative age dating. We know that the rock layers down here at the bottom were put down first, and then these layers were added later, which means that these layers are older while these layers are younger. So if we have fossils in more than one of these layers, then we can say this fossil is younger compared to this fossil. It's a relative age. Whereas absolute age dating gives us an actual age in numbers of years of particular rock layers. It's much better than the estimate that we get from relative age dating. So let's take a look at how we do that. So sometimes absolute age dating is referred to as carbon dating, not this kind, or radiocarbon dating. So let's take a look at how that works. So there are several different isotopes of carbon. So I like to think of the word isotope as flavors of carbon. So just thinking back to your chemistry that you did maybe last year, every type of atom has a specific number of protons and neutrons in its nucleus. And the number of protons corresponds with its number on the periodic table and determines what element it is. And then there can be a varying number of neutrons in the nucleus as well. The number of neutrons will determine which isotope of carbon it is. These are all naturally occurring in the environment, and as organisms eat food and take in carbon and breathe out carbon dioxide, they're bringing in all different types of carbon. So they're incorporated into their body. However, carbon 14 is unstable, which means it's going to decay and lose some of its neutrons over time. So, but carbon 12 is very stable and it will stay the same over time. The way that carbon 14 decays means that it loses half of its atoms every 5,730 years as they decay into another type of atom. So we call this its half-life. Its half-life is 5,730 years. When we look at a fossil, we can compare the amount of carbon 12 to the amount of carbon 14 to determine the age of the specimen. So we look at the ratio between carbon 12 and carbon 14, and that lets us know how long or how many half-lives have passed since the organism had passed away. This only works for specimens up to about 60,000 years old. After that, there's not enough carbon 14 left for us to be able to determine the age of the fossil. But there are other unstable radioactive isotopes of different elements out there that we can use to age other fossils if we feel that they are older than the 60,000 year mark. All right, evidence number two, comparative anatomy. Comparative anatomy lets us look at the anatomy of an organism and compare them to other organisms to see if we think that those organisms have a common ancestry. There are 4 different types of comparative anatomy that we're going to look at: homologous structures, analogous structures, vestigial structures, and embryology. This stuff's kind of one of my favorite things that we talk about. All right, so homologous structures. Homo prefix means same, and we'll come across this in science frequently. So when we talk about homologous structures, we're talking about similar, or in this case, think of homo same structure, different function. So all of these organisms in the image here, the human, the cat, the whale, the bat, they all have the same structures, the same bones present in their forelimb. But those bones help them to do different things, right? So humans grab things, cats walk, whales swim, bats fly. Those are different functions, but they're all made up of the same structure. So when we have organisms that have homologous structures, this means they have a common ancestor. So same structure, different function, common ancestor. Analogous structures, however, are a different structure, but same function. So here we have a bat wing and a fly wing, very different structures that make up the wing. However, both of them have the same function. They allow this organism to fly. But just because they do the same function does not mean that they are common ancestors. We can tell they're not common ancestors because they don't have a common ancestor because they are different structures. They evolved independently of one another. Okay, vestigial structures. I find this one super interesting. So vestigial structures are these parts of anatomy that belong to an organism that no longer serve its original purpose, but give us sort of a window into the past of that organism. So whales, which once walked on land, have these vestigial pelvic bones, these bones that are just left inside their body that used to attach to their hind legs when they walked on all fours on land before they started to go into the ocean and swim. And snakes have these leg bones that even still have a little claw on them that stick out from under their belly that are remnants of the hind legs that they once had in time. These vestigial structures give us a peek into the history of their evolution. Human beings also have an appendix that allowed them, we think, to digest raw meat, which is something that we no longer need. But we still have one, even though it doesn't actually serve a function anymore. All right, last one that we're looking at for comparative anatomy is embryology. When we look at the embryos of very different organisms, so if you see here, we've got a fish, a salamander, a tortoise, a chick, a rabbit, and a man. At the early stages of development, they all are very, very similar, and they have very similar structures. One of which is the gill slits that are found right here in the neck area that develop into different structures as they get older. Obviously, for the fish and the salamander, they will develop into gills. Whereas for the human, a lot of this will become part of the pharynx and the throat and the voice box. So gill slits are one of the 2 specific structures that embryos of very different species have in common. Another one is the tail. You'll see that there are tails on all of these embryos, even though not every one of them will have a tail when they are full grown, like humans. So these similarities indicate common ancestry. So this is one of the pieces of evidence that supports the idea that all living things originated from one common ancestor long, long time ago. Okay, evidence number 3, molecular evidence. So scientists can look at similarities in DNA sequences to allow them to determine which animals are more closely related to each other than others. And this is really helpful because sometimes we have placed organisms next to each other on these trees that show common ancestry. And later, when we look at their DNA sequences, we are surprised to find that they actually are not. Or sometimes the reverse is true. 2 organisms that can look really, really differently. When we look at their DNA sequences, we just find out that they actually are very similar to each other. And then one of the things that we can use is something that we call a molecular clock that allows us to determine how long it's been since two species diverged from one common ancestor. So, evidence for evolution in a nutshell. We can look at the fossil record, which allows us to either use relative age dating, where we compare the ages relative to one another, or we could use absolute age dating with carbon dating to determine how old the organism's fossils are and place them in order so we can see the changes over time. We can look at comparative anatomy. Homologous structures tell us common ancestry, while analogous structures tell us that they do not share a common ancestor. Vestigial structures allow us to see how an organism has changed over time, and embryology allows us to look at similar structures in embryos to determine common ancestry among animals. And then lastly, molecular evidence allows us to look at DNA sequences to determine which organisms are closely related. And we can use something called a molecular clock, which we'll talk more about in class, that allows us to tell how long it's been since two organisms have evolved from a common ancestor. That's it for today. Have a great day.